Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Good afternoon, everyone, and welcome. My name is Kim Ricketts, and I manage, along with Kirsten Wiley, the Microsoft Research Visiting Speaker Series. Thanks for joining us today. Today, we welcome Carl Zimmer here to Microsoft Research to discuss the biography of a microbe. Yes, the story of how E. coli was discovered, and more importantly, the role that this fast-growing and sophisticated bacteria has played in everything from proving Watson and Crick's theory about DNA to helping us understand how genes work to the creation of human insulin. While most of us know it from its deadly outbreaks of undercooked food, in fact, we all carry billions of E. coli in our intestines, and this microbe continues to lead the way in the search for life-saving drugs, clean fuel, and a deeper understanding of our own genetic makeup. When I sent out the announcement for this talk internally, one email came back to me saying, "Yuck." That's what it was titled, and I opened it up, and then he continued on and said, "But upon further reflection, this topic is actually fascinating." And he ends by saying, "Thanks for bringing speakers that broaden our understanding of the world." Carl Zimmer is one of those speakers. Carl is the author of 5 earlier books and is also a frequent contributor to the New York Times, National Geographic, Scientific American, Discover, and as a frequent guest on NPR's Fresh Air and This American Life. Zimmer has been awarded fellowships from the Guggenheim Foundation, the Alfred Sloan Foundation, and his honors include the American Association for the Advancement of Sciences Science Journalism Award, and many, many others. So please join me in welcoming Carl Zimmer to Microsoft Research.
Thanks very much. Can you hear me? Is my mic on? Okay. I suppose most writers would take yuck as harsh criticism, but I am a person who's written an entire book praising parasites, so I take yuck as a badge of honor. I suppose people were not terribly surprised when they found out I was going to be writing about something about like E. coli, and of course, as soon as I would tell them I'm writing this book about E. coli, their minds would jump to getting very sick. It just so happened that there was another outbreak of E. coli when I was working on this. And I would try to explain to them, I would say, no, actually, there's something else I'm after here. For scientists, E. coli means something else entirely. E. coli is really a guide to what it means to be alive. Scientists understand E. coli arguably better than any other species on Earth. And if you want to understand what it means to be alive, then E. coli is a good thing to get to know. So let me just tell you quickly about what E. coli is, and I'll start with its name. Its full name is Escherichia coli, and it's named after Theodor Esterich, this gentleman here. He was a German pediatrician, and in the 1880s he was trying to figure out why it was that all the babies that were his patients were dying. The mortality rate in 19th century Germany was quite staggering. I mean, you'd have to go to a place like Angola today to really find comparable levels of child mortality, and a lot of that was due to intestinal diseases. And so Esterich subscribed to this radical notion that there were germs that were responsible for these diseases. So he was an early adopter of the germ theory of disease. And he thought that he could isolate the germs that made these children sick. And the way that he could do that was, first of all, to figure out all the germs that are in healthy children and then compare them to the sick ones. Because he knew very well that we are full of bacteria, and he wanted to start doing an inventory. So he made a study of diapers, and I'll leave it at that, and tried to figure out what was growing in there. And he was quite intrigued by these little rod-shaped bacteria. He called them Bacillus communis coli, just a common bacteria. And he was impressed by how fast they grew in his lab in the presence of oxygen, and how well they could grow on just about anything. And he describes feeding them milk and potatoes and blood, and they would just grow on all of this stuff. That's basically where things stayed for the next few decades. Scientists came to recognize that E. coli was one of these resident of our guts. You have about a few billion of them. You were infected with E. coli a few days after you were born. You're born sterile, and then your parents or a nurse or someone gets you infected, not just with E. coli, but with maybe 1,000 other species. And they just set up shop inside of you, and you depend on them to help digest your food, to regulate your immune system, all sorts of things. E. coli itself is obviously not a big organism. You could fit about 10,000 of them across your finger. It's got this rod shape, and it's packed with molecules, maybe 50, 60 million molecules. And those little lines that you see are tails called the flagella that I'll get to in a second, because they have an interesting story to tell. Now, the reason that E. coli now is so important to biology is that in the 1930s and 40s, some scientists started to ask, what does it mean to be alive? And they decided to try to answer that question with E. coli. Now, this is obviously a very old question, and Aristotle had his own answer to it. He would say that life, he thought of life in terms of a psyche, that there was something that was a source of movement inside living things, and that non-living things didn't have it. So Ernst Schrödinger in the 20th century, in the 1940s, offered a kind of a new approach to life. So Schrödinger and other physicists were feeling very confident that they had all of physics worked out, but they were a bit puzzled by life, because life seemed to defy physics. How was it that something could give rise to a nearly identical version of itself? And it seemed that it had to do with this strange thing known as genes, but nobody quite knew what genes were. So the reason that these scientists started to study E. coli was because it was harmless, it grew fast in oxygen, it could feed on lots of different things. It was just an easy thing to study. You could start with one E. coli in the afternoon, let it breed overnight, come back in the next day, your experiment's done. And so they would raise them in petri dishes, and they had particular fun getting them sick. So this is a virus of E. coli. And so Max Delbruck and Salvador Luria, two people who won the Nobel Prize for their work on E. coli, would make these poor bacteria very sick. And you'd see, for example, these are viruses that are exploding out of this microbe after it's been infected. So, no one actually knew what the viruses were doing when they landed on E. coli, and then a whole bunch of viruses would come out. Nobody quite understood how that kind of alchemy took place. But by studying E. coli and its viruses, they started to get an understanding of it. So one experiment, for example, what they did was there was this question, well, maybe genes are made of DNA, maybe, or maybe they're made of protein. So what some scientists did was they did one experiment where they basically tagged the protein with radioactive tracers and on the viruses and let them infect E. coli. And then they spun the viruses and E. coli apart. And it turned out that when they looked for the radioactivity, it was outside of the microbe. Then they tagged the DNA, and they did the experiment again. And now when they spun the E. coli and the viruses apart after the infection, the radioactivity was inside of E. coli. And so it was clear proof for those who doubted it that DNA is the stuff of genes. Joshua Lederberg, who just died recently, discovered that E. coli has sex, kind of a weird sex, admittedly, but one microbe will build a tube and pump DNA into another one. So you can throw these E. coli having sex into a Waring blender, which is what some scientists did, and break them apart while they're having sex and discover which genes have made it across and which haven't. And you can interrupt their love life after half an hour or an hour at different time intervals. And you can discover that different genes have made the journey. And that lets you make a genetic map. So one of the first detailed genetic maps came from studying E. coli this way. Another bit of Nobel Prize-winning research with E. coli was the genetic code, figuring out how DNA gets translated into protein. And what was really important about all of this is that once people figured this out with E. coli, then they could go and test these ideas, test these patterns on other species. So it turned out that E. coli had a particular genetic code, and lo and behold, so did we, and so did frogs, and so did mushrooms, so did all life on Earth. So things that were discovered in E. coli kept turning up in other places. Some French researchers discovered a similar thing about how genes are switched on and off. And so Jacques Monod said this wonderful thing: what is true for E. coli is true for the elephant. By 1969, these folks were feeling very, very proud of themselves and very confident. And so Max Delbruck, now here looking much older, declared, this riddle of life has been solved. Now, I think he was getting a little bit ahead of himself. It's not like biology is all screeched to a halt. In fact, there are lots of big open questions about E. coli, about life in general. And E. coli is still at the forefront of dealing with these big questions. One of the reasons is that we know so much now about E. coli. So its entire genome has been sequenced. Actually, about 30 different strains of E. coli have had their genome sequenced. And scientists understand what a lot of those genes do. That's much better than we know with humans. The human genome is really quite a mysterious mess. So one of the things that you need to understand how life works is how the genes interact. Genes don't work in isolation. So there's just one quick example I'll tell you about is how E. coli deals with getting hot. If E. coli gets hot, it could die because these proteins become unfolded and then they're in big trouble. So what E. coli does is it makes an RNA molecule that stays folded up and can't be translated into protein until a certain temperature, and it unfolds, and then it can start being made into a protein and can start switching on other genes. These genes then can take unfolded proteins and fold them back up again, or they can destroy them. But these proteins also destroy this sigma molecule, the thing that started the whole process. So there's a feedback loop built into it. And what's really been interesting is talking to engineers, people who do control systems. For example, I wrote a piece about John Doyle at Caltech, who used to design software for control systems like for the space shuttle. Now he studies E. coli. Because he finds that the way that the whole system works is incredibly elegant and uses a lot of the same principles that he needs when he's designing some system for keeping the space shuttle from flying out of control. So if you're going to be alive, you also need to kind of have at least some sense of where you are. And E. coli actually has exquisite senses. It has this kind of, I think of it as kind of a microbial tongue at its tip. And it's made up of thousands of these receptors, each of which can grab onto certain kinds of molecules, and then it can process that information. It's kind of like a brain. Now, for E. coli, the one big decision is where to go. And the way it makes that decision is that once some molecule latches onto it, there are several molecular reactions that take place inside the bacteria, and that affects those tails I was telling you about, the flagella. So here's a beautiful computer rendering of a flagellum, the base of it, and it has some resemblance to a motor. And what happens is the proteins stream in, and they use the energy to spin around hundreds of times a second. And the little tails, all sort of, they're spiral shaped, and they all kind of bind together if the motor's turning counterclockwise. And that's important, because that's how E. coli gets around. So when they spin counterclockwise, they go in this run, as you can see here. And every second or so, they go clockwise. And they fly apart, and the poor thing goes spinning around. Laser pointers are good for something. And then they start running again. Now you think, well, how are you going to get anywhere with that? Well, if there's something if there's no stimulus, they just kind of wander around and don't get anywhere in particular. But if there's something that attracts them, they just bias their runs and their tumbles. In other words, they run longer than they tumble. And the runs go longer, I should say. And if there's something that repels them, then there's a shorter interval between their tumbles, and once they get pointed in the right direction, they go away, and they get away from what's bad. So it's this wonderfully simple, elegant system for getting E. coli to what it wants to go to and get it away from what it doesn't want to go to. So it can go to food, it can go away from toxins, it can go to the right temperature, it can go to places where there's enough oxygen, and it can take all these signals in and integrate them together at once. It's remarkable. Another important thing about life is that you can't be sloppy about it. And people used to think bacteria were sloppy, that there was just a big sack of molecules and you didn't have to care about how it was organized. Well, that's just silly. Because if you popped E. coli and let the DNA stream out, you would find that it's 1,000 times longer than E. coli itself. Now, if you've got 4,000 or so genes in E. coli, and they all need to be read at lots of different times, you've got to keep it organized. And scientists are just starting to discover the organization that E. coli uses, and it's quite beautiful. And it involves, for example, little clips. They keep little loops in place, and they open so that the gene can be read, and then they close again. And so you can be reading genes to make proteins and copying them at the same time, and you never get tangled up. Another important thing about life that scientists are increasingly appreciating is that when you're alive, you're never alone. You might think of E. coli as just being some isolated, stupid little microbe that just sits there and feeds and divides and feeds and divides, and that's the sum total of its life. The fact is that E. coli talk to each other. They release molecules that are taken up by other E. coli, and the other E. coli change the way they behave in response. And they can do very complicated things. They can, for example, build biofilms together. They'll once they figure out that there are enough of them in a neighborhood, they start squirting out glue and other similar compounds, and not glue, I should say, but glue-like compounds, and will form kind of these microbial cities that line your gut. They will also become altruists. So, for example, if they're running out of food and you've got 2 colonies of E. coli, a few of them in one colony may start to make a toxin called a colicin, and they will essentially like fill up with this colicin, like thousands upon thousands of copies of this until they explode. They kill themselves producing this toxin, which then sprays out. Now, in their own colony, the E. coli don't die because they make an antidote to this toxin. So each E. coli carries a gene for a toxin and for its antidote. But when the toxin goes to the other colony, they die because their antidote is the wrong antidote. They make a different kind of toxin, make a different kind of antidote, they get killed off. And so the colony where those few made that ultimate sacrifice, now they can eat all the food that's left. And this brings us to another important thing about life. I mean, how life, the basic fundamental fact about life is that it evolves. And so this altruism I was telling you about, all these things are the product of evolution. And E. coli is actually a great thing to study to understand how evolution works. There's this, if E. coli is fantastic for how fast it reproduces. I mean, at its fastest, it's about every 20 minutes it can divide. Now, if that just went on, for a few days, you would have a world of E. coli. I mean, we'd be wading through E. coli. But obviously, we're not. The reason is that the resources are limited, and so that creates the conditions for natural selection. So some individuals are going to be able to reproduce better than others. So Darwin had this fundamental insight about evolution, but he was thinking mainly in terms of animals and plants. He really didn't think much at all about bacteria. And yet, E. coli is becoming one of the best things to study to understand how evolution works in real time. And some of the best stuff is coming from Richard Lenski at Michigan State University. So what he did is he started 20 years ago with a single E. coli. And he created 12 lines, genetically identical lines that he kept in flasks. And he gave them a very limited diet of glucose. And every day they'd run out of their glucose, and then in the next morning, he and his colleagues would take a little bit from each flask and put it into a new flask and start it over again. They froze some of the original ancestors in each line, and then every 500 generations they froze some more. So they have this, what he likes to call a frozen fossil record. And then you can thaw them out and see how they're similar and different than their descendants. And you can actually pit the ancestors and the descendants against each other, like in a petri dish, see how fast they reproduce. Now, what's interesting is after 40,000 generations, they're very plump. So they're about, for some weird reason, no one knows why, they're twice as big as their ancestors. And this is a genetic change. This is not some environmental change. And just as Darwin would have expected, mean fitness, in other words, how fast they can reproduce, they are about 75% better than they used to be. And I actually just blogged today about a new paper that just came out where Lenski has discovered that his E. coli have actually discovered a new way to eat. So he feeds them glucose, and he keeps them in this broth that has a mixture of other things, including a molecule called citrate. And one of the defining characteristics of E. coli is that it can't eat citrate. Well, in one of his lines, they're now eating citrate. And he's trying to figure out what happened. How did this essentially, essentially sort of the origin of species has taken place in his lab, and he's trying to figure out what just happened. Why are they eating citrate? Obviously, this has important implications beyond sort of your basic understanding of how evolution works. Right at the time that scientists were using E. coli to understand the basics of biology, of genes and proteins and so on, the first antibiotics were being used. And right at that time, scientists were discovering that E. coli and other species were becoming resistant to those antibiotics. And it was just a classic case of evolution in action, that resistant mutants were being favored. And resistance was spreading through populations of bacteria. And this is an issue even now as we deal with a horrible problem with antibiotic resistance. Now you regularly have bacteria that are resistant to 1, 2, 3, 4 different antibiotics. And so there's this search for new kinds of antibiotics that could trump the bacteria. And one of the most celebrated examples came from frog skin. Frogs produce these peptides, these small molecules that, at least when they were discovered, seemed incredibly effective against bacteria, just lethal. They were discovered because a scientist was cutting open frogs to harvest their eggs and then sewing them back up and then throwing them back in this dirty tank where they were just fine. And he was thinking, wait a minute, these things should be dying of infection, but they're doing fine. And he discovered that their skin was covered with these totally different class of antibiotics. And so everyone said, this is it, because they worked in a totally different way than other antibiotics. It seemed like there was no way that bacteria could evolve resistance to them. So again, there was a great triumphalism until somebody who knew E. coli pretty well published a study where basically what they did was they exposed E. coli to very low levels of this antibiotic, and then higher levels and higher levels and higher levels, and they evolved resistance to this new group of antibiotics without much trouble at all. So the other important thing about life is that not only does it evolve, but it diverges, it diversifies into new kinds of species. And so Darwin drew this tree to show how life evolves. And here's actually where those disease-causing strains of E. coli really come in handy. So I do talk about the E. coli that makes you sick in my book. And I talk about it in the context of the fact that, as I said, we all carry E. coli inside of us from birth to death. And yet there are these strains of E. coli that can make us really sick. They are E. coli. They share thousands of genes with the E. coli that we carry with us. But they do weird stuff. So this is the kind of E. coli that you would get if you ate a bad hamburger. And it's caused the cells in your intestines to grow up into these bizarre pedestals. It's almost like they're building themselves a throne out of your guts. They have lots of different ways of making you sick. There are lots of different strains that make people sick, and the ones that you find in hamburger and spinach actually cause relatively few deaths. The most deaths are caused by other forms, sometimes called Shigella. They kill about 1 million people worldwide, mostly children. So we're not that far from Theodore Estrich's days. And what a lot of them do is they build a needle that they use to inject molecules into your cells, and that causes your cells to act very strangely, in part pouring out all sorts of nutrients that the E. coli can feed on, also causing diarrhea, bleeding, in some cases death. But when scientists look at the relationship between the ones that make us sick and the ones that we carry with us our whole lives, they find that the disease-causing strains have evolved from harmless ones, and vice versa too. So you see that there is this tree here reflected in what we know now about E. coli. And it's continuing to evolve. This is something I wrote about recently in Slate. When there was that outbreak in 2006 of spinach, on contaminated spinach and lettuce, they sequenced it and found that there was a rapidly evolving branch that had produced this particular epidemic. It was different than the other members of its strain, which is called O157H7. So this is actually happening in a weird way, a way that Darwin wouldn't have predicted. What's happening is that, remember I talked about that bacterial sex? What's happening is that bacteria are trading genes. And in some cases, viruses are infecting one cell and then picking up genes and carrying them to another cell. And so you're getting this incredible mixing of genes. It's not just this slow process where a gene might mutate every once in a while from ancestor to descendant. It's really kind of a genetic orgy going on out there. And it's not just going on between E. coli, but E. coli and lots of other species. And this is continuing to happen. The viruses themselves are mutating, so we're going to continue to have these new. We're going to continue to have new strains of E. coli that continue to make us sick, unfortunately. And this gets to. This is important not just medically, but in a very, very deep, profound way. As I mentioned, scientists are sequencing lots of genomes of different strains of E. coli. They're up to 33, or actually more now. And from one gene to another, you will find hundreds upon hundreds of genes that are not found in other strains. So now scientists are talking about how E. coli has sort of a core, sort of an essence of E. coli, you could call it, but it's only about 2,000 genes. And then if you look at all the genes that are found in various strains of E. coli, they're up to almost 10,000. And they expect they'll probably get up to maybe 20,000. That's as many genes as we have. And so scientists are now talking about the pan-genome. This is going to be a phrase you'll be hearing a lot in the future. So life branches like a tree, sort of. You do have a tree growing through time, but then you have all of this transfer of genes between the branches. And so this is a profound thing about how life works, and E. coli is really important for revealing it. Just very quickly, I'll tell you a little bit about how E. coli became a creationist poster child. This is how intelligent design people like to think about E. coli and its flagellum. They think of it as a motor, and they say if it looks designed, maybe it is. And if you'd like, you can order this on a throw pillow or an apron or a beer stein. It's up to you, and there's where you can get it. And creationists for many years have been likening E. coli's flagellum to technology of various sorts. And they put it on the covers of their books. But this all came to a head a couple years ago in this famous case where a board of education in Pennsylvania was trying to get intelligent design, the newest flavor of creationism, into schools. And one lawyer joked that you should really call it the flagellum trial, because a lot of it revolved around this flagellum, and particularly of E. coli, since E. coli is the best studied flagellum of all. One of the arguments that creationists make is that something like the flagellum couldn't have evolved, because if you take away one part of it, it doesn't work. So you would have no earlier forms of the flagellum that could evolve to a full-blown flagellum. But this just doesn't make sense, because let's say we've got E. coli's flagellum, and let's take away that long sort of tail part of it, and take away the hook, take away these other parts as well. Let's just take away a whole bunch of it. Are you left with something that works, something that has a function? Well, you are. And actually you can find these things in other bacteria. This is a close relative of E. coli called Buchnera, which only lives inside aphids. And it only lives inside the cells of aphids, and it's essential for the aphids. It generates some of the food they need. And it's got protein for protein, this part of the flagellum, and it uses it probably to release molecules into its host cell. So E. coli not only doesn't support the creationism, but it gives you an idea of how evolution works, how complex traits evolve. So scientists are now looking at all the parts of E. coli's flagellum and finding homologies, related versions in other structures in bacteria. And just finally, I want to talk about another reason that E. coli is such a great thing to study and to use to learn about life, because life is changing. I mean, we live in an age of biotechnology, in an age of synthetic biology, and E. coli helped us get into this new age. And I'm particularly interested in what E. coli can do to help us to understand what's natural and what's not natural. Because a lot of the debate these days about genetically modified foods revolves around, or chimeras or so on. People are nervous about it because you're crossing the species boundary. This is Marlon Brando in The Island of Dr. Moreau, and he meets a pretty awful end because he's tampering with that species boundary. H.G. Wells punished him for doing that. Well, in the early 70s, scientists figured out how to put human genes, I should say, they figured out how to put animal genes into E. coli. And they started out, one of the earliest things they did was they put some frog DNA into E. coli. And once they had figured that out, then they thought, well, we can put other things in, like human insulin, for example. This generated a huge controversy, just as intense as what we're dealing with today with genetically modified foods and so on. And it's really interesting to go back and look at how that played out. People were really scared that E. coli would be killing people left and right. You would have E. coli delivered plagues of cancer or diabetic shock. And the city of Cambridge stopped all genetic engineering for a few months because there was such an outcry. Today, things have changed a bit. So while genetically modified foods may be controversial, the fact that a lot of insulin comes from E. coli isn't, which I find interesting, because this is just as unnatural and a breaking of species boundaries as a mouse with human neurons in it. We've actually gone a long way from those early days of genetic engineering. Now we're in the age of synthetic biology, and it's E. coli again that's playing a big part. And right now we're kind of in the we're still, I think, in the kind of cool stunt phase of synthetic biology. So some undergraduates and graduate students figured out how to engineer E. coli with some pigment genes from marine bacteria and some other genes, and basically turned it into a camera. You can take your picture with E. coli. But there are some much more ambitious projects underway. The University of California at San Francisco, some researchers want to engineer E. coli to basically home in on cancer, invade tumors, and then once they're inside, release a bunch of toxins. And now E. coli is producing gasoline and jet fuel, malaria drugs. It's been used with. What I find interesting about this is that it not only has some real applications in the sort of commercial world, but it might help us to answer some of these deep questions that I was talking about before. How far you can tinker with E. coli might tell you a lot about what the limits of life are. NASA is very interested in this question because if they're going to send probes to look for life on other planets, they'd like to have kind of an idea of what they should be looking for. I mean, is it going to be like Star Trek, where you go there and, oh, look, there are people on two legs that just happen to have green paint on their faces? Or will they be different? Does life have to be made of DNA, for example? I mean, all life on Earth uses about 20 amino acids to build proteins. But there are dozens and dozens of other amino acids they could use, theoretically. Is there something wrong with those amino acids? Do they not allow life to exist? Well, what some scientists have done is they've engineered E. coli to use some of these unnatural amino acids to make their proteins. They're fine. They just go on with their lives. They've used 30 different amino acids now that, as far as we know, nothing on Earth has ever used. At least we don't know of anything that's used them. So in a sense, we have made aliens on Earth. They just happen to be E. coli. So the question that I have, if and when we ever find life on other planets, is to take Jacques Monod's saying, what is true for E. coli is true for the elephant. I want to know, is what is true for E. coli true for aliens? I mean, E. coli is already up in space. I mean, it's in the space station. It's in the astronauts, and it's floating around in the air there. I don't think that when people go to Mars that they will actually find E. coli. But I do wonder if knowing about E. coli will help us to understand what they do find. So, thank you so much for coming to my talk. Yes, sir.
Is the motor really going 360 over again around to get the E. coli moving? Is it a wheel?
Yeah, it goes all the way around.
Over and over again?
Through 100, I forget the figure. I think it's about 200 or 300 times a second.
Okay.
Yeah, there's been a lot of, over the years, like biologists have made a big deal. I know Stephen Jay Gould would talk about how life has never evolved wheels, like there are no animals with wheels, and talking about that as some sort of constraint, an example of a constraint on how life can evolve. And I'm thinking, well, what are you talking about? I mean, the animal kingdom's all well and good, but in the bacterial world, there are wheels all over the place.
So you can't have blood vessels going into
Right.
a wheel. That would be hard.
Yeah, but there are definitely wheels or wheel-like things on E. coli, and they spin around 360 degrees going counterclockwise, and then they stop and they spin the other way 360 degrees hundreds of times a second, and back and forth and back and forth. In the back? I'm sorry?
The E. coli that have the extra proteins that we don't normally have those been found to be dangerous, or are they so divorced from our biology that they wouldn't infect us with anything bad?
Now, are you talking about the ones that are using unnatural amino acids? I don't think anybody knows. But people are thinking about medical applications for those proteins, and because they might interact with our bodies in interesting ways. Hopefully in good ways, but there is medical research going on about how to use these unnatural proteins. Sort of a variation on that is so when our cells make proteins, once the basic chain of amino acids is made, they are then further processed, and in some cases, different compounds are added on to some of the amino acids. Normally, bacteria can't do that. They don't have that equipment. So what some scientists have been doing is saying, well, maybe we can engineer E. coli to take amino acids that already have some of those extra molecules added to them and put them together, so that you can still get E. coli to make these proteins that have these little additions on them. So it's a different way to get E. coli to make human proteins by just changing its genetic code.
Crick Venter's talking about trying to create bacterial life, I think, from scratch. How much more complicated is E. coli than what he's working toward?
So Craig Venter is working on a species called Mycoplasma genitalium, which has one of the smallest genomes known. I think it's maybe 500 or 600 genes. But what he's been working on is, he and his colleagues have been working on, is figuring out, of those genes, what are the bare minimum that it would need to survive. And I think they're down to maybe 350 or something like that. Now, what they do is they just knock out one gene at a time and see if the thing still lives. Now, to really know whether that's the minimal set of genes, they'd have to recreate it with just those 350 genes from scratch and see if it boots up. And nobody knows if it would or not. But he's taken so many steps now towards that goal that a lot of people, within months maybe, he might have something like that. Some other people have suggested using E. coli instead, only because it's so well studied. I mean, there are a bunch of genes in mycoplasma that people just don't know what they do. They're essential, but we don't know what they do, which is surprising. With E. coli, scientists know a lot more about all those genes, and so George Church at Harvard and some other researchers have said, here are I think it's about just 200 genes that he thinks from E. coli you could use to make something live, the minimal set of genes. Once you figured that out, then maybe you could just build up on that chassis and create things that are very different from any bacteria known today. That's the thinking. I mean, other people say, why bother? Because there's so many bacteria out in the world that are so well adapted to doing so many different things. What we need to do is we need to be searching for those natural experiments rather than trying to start one from scratch on our own. Yes, sir?
Do you think this could help solve the problem of biologically degrading plastic? We have so many plastic proteins which are just sitting there because no bacteria can digest them.
Well,
do you think E. coli can be evolved into this?
We may not need to engineer E. coli. There was actually a story that just came out a few days ago. Some kid, I believe in Canada, for a science fair project thought maybe I could find some bacteria that break down plastic bags. And so he just went and looked in some soil and and found a species belonging to a group called Pseudomonas that loves eating plastic bags. And this is actually like a big discovery. I mean, it blew a lot of microbiologists away. This thing will just feast on plastic bags. And so I mean, this could be a very rich kid someday, you know, seriously. Once scientists could identify what it is that this bacteria is doing, some might transfer some of those genes to E. coli, combine them with other genes to optimize what it's doing. That could happen. Breaking down plastic is one of the applications, generating energy in less harmful ways than we do today. That's another possibility. Craig Venter likes to talk about taming our habit of spewing carbon dioxide in the atmosphere, drawing them down with carbon dioxide guzzling bacteria. That naturally raises a question like, okay, what happens if some of those bacteria get out of Venter's grasp and just start sucking down carbon dioxide uncontrollably? Would we suddenly find ourselves in an ice house instead of a greenhouse? I'm being a little flippant, but the point is that there are a lot of issues that have to be dealt with synthetic biology. The bacteria have a habit of slipping away. That you can't. It's not exactly a lock and key that you can use. So scientists are trying to find out ways to basically make it impossible for them to live outside of certain very constrained conditions. So if they don't get some particular kind of food, they can't survive. They'll just die. But then the question becomes, well, what if they had enough time to pass on some of their genes in the way I was telling you about to other bacteria in the environment? What would happen then? Who knows?
Well, then the questions you can, of course, ask Carl them as he stays here a little bit longer in science books. Viewers says hello, but thank you very much.
No, thank you. Thanks very much.